An honor to introduce our next Beltrami Prize award today, and this is Willi Jäger. So let us.
Thank you.
And I will say a couple of words to introduce Professor Jäger. Of course, he's a famous scientist, and everybody knows him. And during many years, Willi was professor at the University of Heidelberg. He had an enormous activity at the university level, at the national level in Germany, also international level. He was a founding director of the Center for Scientific Computing, coordinator of the Center of Modern Simulations, chairman of Oberwolfach Society, and many, many other things. It would be impossible to list them here. But of course, what is also very important, and was also mentioned here today, Willi had more than 100 PhD students. Can you imagine that? Is it true?
It's true, but I didn't count them. That's very important to tell. And on the other hand, I share the opinion of my colleague Preziosi. The most important are our students. And one aim of a professor should be that the students are finally better than himself. And I hope we succeeded.
And also, Willi has several honorary doctorates, different awards, in particular Order of Merit of the Federal Republic of Germany, and many other things. Again, it would be impossible to list them all today. But what I would like to add, and because I know Willi for many years, that he has this enormous passion for science, and what we witnessed during this conference with this interest to all talks, activities, and also this very wide and very broad interest maybe in all sciences, especially in mathematical biology. So it's a pleasure for me to announce again this award and to invite Willi for his lecture. And this was done yesterday during our excursion on the top of the hill. And I would say that Willi was really very good at climbing this this mountain. So please, Willi.
No, thank you very much. I also need, that's a computer. Is it on it?
Let me connect this one. So this one is connected. I don't know where is your...
Where is the...
I know, sorry.
It's here. Therefore I have to wait. Maybe I can already say something before, because I'm very happy to get this honorable prize. Especially I'm very happy to get it together with Professor Preziosi, where I had not a chance to meet him very often, but I know his work and his contributions, and I hope that we can start some more discussions. And I think both have the same aim. We want to that better connection between, including in these fields of mathematical biology and in other fields, a combination of fluid dynamics, reaction diffusion systems, transport with mechanics, which is very, very important to couple all together. The complexity increases, and we have to, my approach is usually that not start with a simple one, but start with a complex system and cut it down to a simple one, in order that one doesn't forget what one has done, what one has assumed. And I enjoy very much this atmosphere of this workshop. And it is an experiment, and I have to tell I'm very impressed by this cooperation already, which has started, and we should really try to support it furthermore. Now, I also want to say in the first page, first of all, let me just say why did I choose this title? This topic, because I don't want to be fashionable, but we are studying for several years already, 7 or 8 years, sepsis, which is one of the challenges of medicine also sciences, which we see. And it's clear to me for already a long time, COVID, if you get this sickness in a severe state, it's developing as a sepsis. And therefore, a lot of things which we have studied there in the case of sepsis can be specialized. You find it in the COVID because COVID is a virus sepsis, if it's in the serious state. And I thought it opens up a lot of questions to mathematics and also especially to mechanics, and take it as a chance in such a, and to maybe hope that some of the younger people try to join this research, which is just not just COVID, but in general, problems of physiology which are getting important in this disease, development of disease. Now let me also say something to Beltrami, because I am related to a certain sense to Beltrami, because Laplace-Beltrami operator has been an important part of my life. In fact, I got my PhD study was the spectral analysis of the Laplace operator in generalization. I did it with variable coefficients. Then I had already a Laplace-Beltrami operator, and I very highly estimated Beltrami. In general, I have to make a very great compliment to the analysis tradition of the very strong analysis in Italy. And we all were very much influenced by the results. Now you know about Beltrami, and especially he was one of the people who were connected also with German scientists starting the non-Euclidean geometry, and he contributed a lot in cooperation with Riemann and Dirichlet and Gauss, and therefore I'm very honored to get this prize. And it's really, Beltrami is one of the people who really explains, if you have such predecessors, you can understand how mathematics could develop under good political conditions very well to outstanding results. And I also know that you recently had some problems, especially for younger people, which were obstacles to continue in this way. I hope that doing this research together will help to get better situation here too. Now, in fact, my partner initially in Italy was Mariano Giaquinta. If you know him, what he does, I was more in the differential geometric side than in this application side. Now, my question is, can we do with mathematics and computational science in more general than just mechanics, also including computational chemistry, to overcome corona and take precautions for future epidemics not to develop to a pandemic? I think it's necessary to use mathematics and computational science. Without that, I think we have a mission impossible. We have to understand the processes, and some people believe that everything can be done now with machine learning. But I advise you, look at the contribution which were done in machine learning to the COVID. And I almost can state almost nothing. In some cases, I don't blame them, because you need data for that. If you have a situation where I have no data, what can you do? You cannot do machine learning in this case. Also, you have to be careful. If you do machine learning and get some result in one hospital, it may not be true for the other hospital either. But we should think, and it's very important for me to mention this, we should not just be process-oriented or data-oriented research, also including the statistics of the data. It has to be combined. We only have a chance to get to the results if we combine it in the correct way, and we should try to help to do this. We have a problem. We have since several years, it started 2015, a joint project with the intensive care medicine faculty, Mannheim faculty. We have two faculties of medicine and University of Heidelberg, included a lot of data. We have a lot of data on sepsis. And as you see, the title says it already, what we had in mind, Scientific Computing for Improved Diagnosis and Therapy of Sepsis. I will take the chance to criticize this project initially already, because it's not just necessary. They had the idea, we have to know, does he have sepsis or not? That's not sufficient, because if you want to cure it, you have to know what type of pathogen is it, and where is it sitting. And I said immediately when I discussed with my colleagues, I would not grant it our projects because it has no microbiology in it. And it does not help if you know someone has sepsis, you take some antibiotica, but you do not know where the source of infection is and what type. And very often people die because of this fact. I can tell you a very story now afterwards where it really happened that some minutes may decide if someone survives. If you do not know what type of infection it is and where the infection is located, no help. And a lot of people died because of this. And this is what I take this experience from sepsis studies, also for the approach to COVID. Now, let me see. It has been, I want to tell what is a sepsis, because in German one said Blutvergiftung, by which is a nonsense. Even the medical doctor now still keeps the name. And this definition which I give is not too old. It was not even existing 2015, it only arose 2017. It's a life-threatening organ dysfunction due to a disturbed reaction of the body to an infection. That's rather broad now, but I think it's necessary because of this. It's a dysfunction of the response of the body with the immune system. Before, they just allowed to have bacterial infection in it, which makes a nonsense. It's a nonsense, I immediately told them, but you couldn't change it up to they changed this definition. Aim of this lecture is to present the critical issues involved in the scientific analysis of the dynamics and consequence of the infection leading to life-threatening sepsis, their description and treatment. To present some concepts and methods and digital technology of mathematics and information sciences, discussion of the transfer of research results into measures for control of the spread of the infection. I will not go to the spread of infection in a population. It would cover another series of lectures, but already a problem is how does an infection spread in a body? And in fact, one of the few contributions, Vitaly, you gave one of the few contributions to this. Almost nothing is done on that. We do not know the virus, even we do not know how the virus load is in the body, because it's not just in the lung or in the nose, it's all over spread there. And already now you can ask, where is it? Where is it? If you know that a virus needs a place to bind, and the place is usually there where ACE2 is located, then ACE2 is usually people who have high blood pressure know what it is. It's related to a control receptor of controlling high blood. And if you know where this is represented in the body, you know where the virus can attach, only there. There are some other possibilities, but they are more rare. And therefore, we are just looking at processes in the body, and maybe we do not look at the spread in populations at the moment, not in this lecture. COVID as a sepsis. I have been hesitating to give you a distribution of numbers. It was, I took it in I had collected it on global wide in October already, but you see the total number of at that time of deaths in the world was 6 million, more than 6.5 million people. But if you look at sepsis, I have that it should be done downward, worldwide. Where do I have it here? Do I have the global for sepsis? But it's higher. It's higher. Let me just go to the German numbers. We had already 2016, 70,000 deaths because of sepsis. And approximately now, in the recently, because you have to always to see, the deaths which are counted in COVID are about 90,000 in Germany per year. It's not so dramatic different. In fact, why I put these numbers here, which you can see, is you have to be very careful. Because how can you separate this organ breakdown from caused mainly initiated by bacteria and finally caused by COVID, or caused by COVID, and then the bacteria took over. Therefore, it's very problematic. This data facts are very problematic. Why I put these figures? You see, you have to be very careful to evaluate these numbers, and I think we have to have a more global view. You have to a more integrated view. I do not know how they differentiate. For instance, the septic death and the COVID. You cannot differentiate that. That's very important to evaluate what one hears. From the point of view of our sepsis research, processes of COVID run accordingly to comparable patterns and can be classified accordingly in relation to the system. It's one of the first virus patterns, virus epidemics, which have been studied now under the aspect of the sepsis effects. Mathematical modeling, analysis, and simulation of the observed are initially in a hypothesis on the process of the level of cells, physiological units, and organs. And if we do that, we do not claim that we really can explain them, the process, but we can better organize research on that and maybe help to develop relevant research for the dynamics of the virus and the viral sepsis, and makes them both more predictable. It is very important to tell that we do not believe that of the correctness of the models, but the models should help us to get better knowledge about the processes. Data analysis with IT methods, machine learning are integrated with the process-oriented mathematical modeling and simulation. We had it in our project. We did that, but the question which was studied in the beginning was a wrong question. The medical people want just to know, do they have sepsis? Have they a chance to get sepsis? And they didn't ask what kind of sepsis is and what kind of development of the disease in a special patient could happen. Therefore, I try to convince them to get rid of this in time single time fact that we said now he has sepsis and then it's finished. No, they should analyze the data and try to classify the different dynamic directions they may have. You have a lot of different ways. Do you have different patients? They both all these patients share that they have a COVID sepsis, but it goes in a different way. And in order to have this would help us a lot if they could do that by data analysis. Say we have about, maybe say, 5 types of evolution of this breakdown of the system. And there we could use the results of which can could get by IT technology and data analysis for our modeling too, because we would have to show that our model shows this kind of dynamics as a possibility. This interaction is absolutely necessary, but it doesn't exist. Even after 6 years, we don't get the answers from that. They give us an answer and tell you, if you find, for instance, they did, one of the first results was lactic acid is not important for indicator, is no index for COVID. When I read this, I said it's nonsense. I can tell you why, by physiological process, because it shows that the oxidization which produces ATP doesn't work via glucoses, because it has been an anaerobic process of ATP production, and then you have lactic acid, and it does harm. And we studied that. But it comes from machine learning. It was classified as not important. Then they had to look it up and came after 3 weeks, oh, now we have a different number. I didn't know how they did it, but they said it at least. Challenges to interdisciplinary research. Let me just see. Is identification typical characteristics of the pathogens and the infection. Mathematical modeling and analysis of their dynamics and control in tests. The development, production, and administration of vaccines and drugs. We were involved with, especially with the tests and the vaccine development, and they came and asked us, what should we do? But with not a very promising results, it didn't listen finally to us what we said suggested. Description of basic structures of the program of the virus, analysis of dynamics of the virus in the host at the cellular level and in the system of organs. We have to understand what on the cellular level happens. We also have to understand what, for instance, find some indices which characterize the dynamics of the virus and spread the virus in the host. And one factor which is very important, we should see, if we would know it, we would be very happy. And one of our young colleagues said, when I'm asking what are you studying? I'm studying a virus load. Virus load. If I would have a reasonable definition and accessibility of virus load, would be great. But we can't. I don't see how to do this, because we do not know how the virus spreads and where I should look measurements on that. Then mathematical modeling and analysis simulation COVID as a virus, sepsis. Here I put already important part, hypoxia, inflammation, impaired immune responses and their consequences. If you look at COVID, in the first part of the COVID, if they would have asked us, the doctors would have asked us, we would have, you would have told them directly, look at infarcts and look at thrombosis. But they did not look carefully. Therefore, a lot of people initially died, not in the second wave, because they knew what to do. But if they would have asked us before, we would have told them, because hypoxia immediately influences thrombosis of. And then there you have hypoxia will become clear, because one of the most attacks is in the lung, which we still will discuss later. There, and if you have the lung, is very important for oxygen supply, but also for getting rid of CO2, which is very often forgotten. If it stays, the CO2 just stays in you, because it cannot be transported out as it should be. It has effects also on the local physiology. You have two kind of reactions: super inflammation, or what is also possible, totally immune suppression. The patient can react differently. And why is this the case? To try to understand it. Supercoagulation, I mentioned that already. It could have been predicted by us. We could have told them. We did, but we had no influence. Enhanced plaque formation, thrombosis, multiple micro-macro infarcts. Acute organ system damage and long-term consequences. Now, this is a very important topic. And in fact, it really is a challenge, not just for the pandemic, but the aftermath which we have. Sorry if I take a little bit too long afterwards, please excuse. I will tell you by example. I had very good relations to our president of the lung specialists in Germany. I thought talking a bit about him, what does the virus do to the lung, would be the correct topic. No, he was no longer interested. He was more interested, how do we have such long COVID phenomena? Because it's mainly damaging the microvascular system. And there is a new topic now to look how did it come up and how can it be repaired, because people who have this suffer a long time and it's an aftermath, and there he is now not so much interested in the development of the infection in the lung, but he is interested how can we help who had this disease. Okay. And I already said I will not go in the other part. Now you see already why the start usually we assume that the start is in the lung. It must not be like this. It could be also some other places to get the COVID in. And here what you see on this first up here on the left-hand side, so is the bronchial part. Vitali was talking about the lower part. And we are concentrating on the alveoli. And because there we are interested how will be how important are they for the exchange of the gas exchange. And so how can we imagine what happens in this highly complex system, and we cannot really approach this as we should, because we are not able, essentially the capillary system is really a terrible problem to describe mathematically correctly, and we have to work on that. Therefore we always assume small vessels where we still could apply some kind of differential equation to describe the transport there. But it's an open problem how we could make it improve. So virus attacks the walls in the lung, destroys mainly the endothelial cells and the epithelial cells of the alveoli, and impairs the oxygen supply to the organism and the release of CO2. Hypoxia automatically comes up, leads to inflammation in the lungs and in the whole body in functional, and they create really a disturbance of the whole system, in the functional disturbance of the whole system. The body reacts immediately. In each hypoxia, it produces some factor which is called HIF. Hypoxia-induced factor, and tries to use it to control what happens. But this is then done by a lot of signaling, and this disturbs the normal situation, and it can be really finally confuse. No? For instance, when I said the neutrophils cannot do chemotaxis anymore, why it could also become they got too many messages in which direction to go. Because they can no longer differentiate the information, because they have much information, or if they could just get immobile or less mobile, or by ROS production and by the defects coming from the hypoxia. Then also the waste of all these damages will go through the whole body, and finally, in fact, and we are especially interested in such structures. It's already one structure which you see there, which we strongly think it's very important. We have to look at the interfaces between compartments. The interfaces, here is the interfaces of blood and of the blood system, and the interface with the air. With the bronchial system. And in other situations too, you can have it, as we will see later, in other organs too. These interfaces are very important, and we have to try to show what happens at the interfaces, at how we get the transfer or coupling of the system between which are valid in both sides, in order to see what goes on. Now, I hear you know everything about a virus. But we have to know what are the important parts. I am always astonished that all the genetic information is stored in the inside, but it's not large. It's not large. But it creates a lot of problems to us. But the more interesting part for us is also the spike glycoprotein, because the spike glycoprotein is that where the virus hooks on the cells, and it only can proliferate, as we know, not outside. It's just like a dust particle. It's not can be not replicate. The replication has to be inside the cell, using also system of the cell, and therefore. But we have to look especially also on the spike glycoprotein, and therefore let's look a little bit at the cycle. Here you see how it hooks on, and here I told you already that it's very important that it only can hook on via this spike glycoprotein, and we have also spike glycoprotein. We have two parts. It's one part S1, that's where it hooks on, and one the S2 part, which is on the virus, closer to the virus. This is responsible for the transition through the membrane to the cell. And for instance, it's now the new kind of versions of the virus. They have changes mainly in S2 and not so much in S1, but there they can better penetrate also. And the movability has been shown also by mathematicians. Bob Penner, who is a specialist in studying this spike glycoprotein. Now let me go. I just want to say, this is very important too, to realize this. Patients suffering from hypertension have more problems with the virus. Why? Because they have more ACE2. And our first idea, you try to get rid of the ACE2. But if you do some drug which takes this off, then the body produces even more. Therefore, the people who have a lot of hypertensions are very exposed to the. I have to hurry up because otherwise. Here you see it again. The ACE binding the spike glycoprotein, and if you look at this, and you know where it should hook in, you see a hole up there in the structure. You could see visually there why it couldn't hook in, and in fact this can open up. One knows this. And here you have the picture of angiotensin ACE2. It has essentially the part which you can put in. Therefore, just doing by graphics, you have already guessed, but that's not enough. You have to do it you should do it by molecular dynamics, and also should do it respecting the mechanics of these problems, because it's not. I show you this by picture. You can really try to put it here in, but it should be done by. The simulations are not existing yet. If you would understand the mechanics of this system better, and how you could block these mechanics, you would already have a good drug to stop this. Therefore, in the discussions also with Bob Penner, I always tell him, what you do is very interesting, but we would like to have done this by molecular dynamics, because this system cannot move as it wants. These modules which you can have in movement are very closely related to mathematics. I don't want you to understand that, but there is a relationship of these possible configurations, and I want to say this. No, wait a moment. Is it already here? No. Did I cancel it? No, probably I can't do it. It's very closely related to the class of Riemannian manifolds. And therefore you can use also abstract theory to see what you can do. What are the individual stages which have to be looked at? You have all this docking, access, intrusion, release, translation, replication, construction of the new virus material, extraction of this virus material, putting it in a form that it can be transformed out to the body again, to the exterior of the cell. And all these processes should be mathematically modeled to a certain extent in order to understand that. Especially the replication, because if you, let me also see, we did also something, interaction with membranes. I show you some picture here. Here you see the microscopic situation at the membrane. You can guess where these dark parts are the are the viruses. And here you see up the development of some the deformation which is done with a cell by them. And here I show you below down some images how this budding comes up, which we did not do it for the COVID, but we did it for HIV and hepatitis virus. How does the virus manage to go in the cell, penetrate in the cell, and we set up a system, putting up some for the mechanics of the membrane, also included some chemical description of chemistry agents. We use the Canhilliard approach, too simple approach for that, and in order to find out can we simulate that such a virus, by using chemicals, change the situation of the membrane, that it can have this budding and finally move in. And we did this for the hepatitis and HIV virus in another program. And down here you see the Helfrich functional, which includes the curvatures, the mean curvature and the Gaussian curvature with a factor which depends on the chemistry phi, and on the right hand side you have this usually the part which reflects the chemical energy, and then we had to derive from that, we had to derive the evolution down to minimum, and this gives rise in the limit to a very complicated system of it's fourth order in space derivatives, also for the part of chemical part, and this is so general that we do not know the existence of that, but you had to compute it. And it's a generalization of the so-called Wilmer equation, which just is only a part of the first part. But we are advanced in computationally, it's okay, but a lot of theory would be needed to show even existence for those things. I hope that at least it wait a moment, I should try if it, wait a moment. Where is it now? Sorry, I wanted to go back because, let me see, because it's dynamic here. It should work. Oh, it doesn't work, sorry. It's very, also numerically very interesting. So we also did then, not for the COVID, but for the HIV virus, where we have no vaccine. But therefore we were very much interested where are checkpoints, where if you find in the dynamics a point, if you stop this process there, then you stop the development of the disease. Okay. I go over now for the production of antigens. let me just mention the state of the, which would be highly necessary to study, is what is the processes of, if you have antibodies, how is the production of if you have production of antigens, how is the production of the antibodies? We are at a state, at the level of George Bell, I think, if you would agree. It's not very much done that, because we would really know what kind, under what conditions the immune globulins are built. And it's a new field of research which should be done, and I only can ask young people to get in this field and make this gap, fill this gap which we have now for more than 20 years. Hypoxia as a determining factor, and I told it already, therefore I can a little bit hurry up. And I also said to you, hypoxia is controlled by the body by a production of this HIF factor, and this controls everything what could help to improve the situation. One could be kill the cells. One could be generate new vessel systems to get better supply, or try to influence the balance of different kind of oxygen production. Is one of the central parts of the system, which we study very carefully how it acts on the dynamics in the supply of energy. And I show this now in a picture. Because my time is running out a little bit, you see this hypoxia. Essentially, I look at hypoxia. Hypoxia can have an inflammatory effect, but also it can be an anti-inflammatory effect. It depends on the state of the dynamics. Therefore, usually people do not observe that it can be. It's both in it, and it's on the level of the cells. It's a blue part. Then on the molecular part, on the intracellular part, and so on the level of systems of cells, and the level of organs. We have metabolic changes, cellular structural changes, mismatch cellular energy demand and supply. But it's very important are the mitochondrial dysfunctions, because mitochondria are really important for the energy and substance supply. And changing of signaling, changing in cellular differentiation, motility, adherence, apoptosis, functionality. Functionality is an important factor because it influences finally on the organ level the dynamics. Epithelial dysfunction, vascular dysfunction, alteration in hormone supply, activation of coagulation, dysregulation of the supply network, dysregulation of the immune system. And what we are looking at is essentially we want to find vicious cycle. That means if you start with one process, it automatically gets you down to the point which you would not like to get. This detection, where are vicious cycle which we have to observe. And now I show you one. We have to study what happens in the metabolism system of mitochondria. I just show you a network of ATP production. One is based on glucose, and the other one is on fatty acids. But we also have other basic substances which have to be controlled. And all this stuff here is in a huge network controlled by this HIF factor. And we did a very careful analysis on that system. And down here, you see byproducts, which are very dangerous because this ROS production influences, for instance, the property of the environment. And here we have one situation which we also were very much interested together with Adelia and our partners in studying the inflammation process, how the immune system reacts, how monocytes are transported to the places where they should be, where they should help to fight the sickness. And it's a problem. You have the blood flow, you have the monocytes, they have to pass inside the vessel wall. And what happens in the vessel wall, how do they change the vessel wall, and what is the effect to the vessel wall properties. We studied in a joint research work, which I will show you in a second, in a solution of a PDE system. Here what we did, we have the Navier-Stokes flow in the blood flow, and then we have a BO equation for the pore-elastic vessel wall. We have a convection chemotaxis of substances and of immune cells. We have done reaction diffusion equation for transport signaling and interaction processes that trigger inflammation, and with a focus on the role on the endothelial layer and the intima. And we have also growth modeling in the intima, which is caused by that the macrophages get foam cells, which have a larger size. And one important factor in the analysis was the shear stress. We had already some discussion in importance of shear stress, but the shear stress in those processes is very important. And we did simulations on that. I also Adelia, we did it also for the PO in the meantime, but only for short time, because for long time effects which take a long period, we have instabilities in the algorithm. We cannot do it we only can do it for short time. What you see here, we start initially with the blood is above this blue bar. And here is down, we start with a curvature because we want to see what the influence of the shear stress is. And then we have treated everything together, what we could do, also chemotaxis. And you have what are important factors? So low density lipoprotein, which is located at the endothelial layer, and its oxidized version is in the vessel wall, and this oxidized version is influencing the permeability of the endothelial layer. And therefore, we had to put them in because we want to see how in the dynamics devolves that the monocytes can go from the blood through this endothelial layer inside and change then to macrophages. And then build this formation of and here you see, you have here, oh wait a moment, now I should go back. Here you see evolution in time, and for all this different kind. On the left-hand side you have LDL, on the right-hand oxidized LDL. It's a different stages. You have the monocytes, how they evolve. And what is important, you see a hump coming up there. It comes from the growth. The first hump is by curvature, which is restricted. And that's, we would like to see if it mechanically may break up and disrupt and get to thrombosis. So then let me show you one thing which I want to, maybe I should then finish with essentially with after some, I will not do some contribution to analyze this layer system because time doesn't allow it. But what I want to tell you is we are looking for this vicious cycle. And one of these vicious cycles, I think, is the following. What is important for the transport in the organ system? It's a heart. The heart needs to pump. To pump needs from the muscle cells, needs the energy supply, and the energy supply is coming via the mitochondrial activity and ATP production. And since the muscle cells muscle cells are very challenged by their activity. It's not enough to have glucose transformed to ATP. It also needs to have fatty acids. And these fatty acids need, in order that they can be produced to energy out of it, need some other substances. One of them is wait a moment, do I have it here? It's creatine. Where is it written? No, it's not written there. It's carnitine and creatine. They are formed, some in the kidney, some in the liver, and they have to be transported. Now, if muscle cells are not supplied well enough, then they pump less. Then the transport coming from the kidney or the liver is even going down further. That means you already see starting a cycle. And now comes an effect which I think we have had it before. The muscle cells, the cells react. They go on strike. They get quiescence. And if you get quiescent, then you you no longer contract. And that means what you do finally, the pumping goes down more and more. But it's vicious because the supply also gets down. Then it may go down finally totally. And here, I think we are working on this to have this modeling and try to compute this situation. Now I only will show you finally in some pictures where you can see where we have similar situations, not just in these situations of layers, which are important to know what the activity is there during evolution of this disease. It's one thing we saw already before. Wait a moment. The layer problems. They come up all over, and our study is mainly now mathematical studying of such layers, and how can we describe the transition. We saw already this layer in the situation of the blood vessel. You have the moving up still the moving monocytes, which have to penetrate. There you have it also in the gut. The gut also is a very important part of a layer where we have this transfer to describe this, and there's very little mathematics done on it. Avner Friedman did something on it, but it's not sufficient for our research. And then we have here what you saw already before, and this we not just look here in the images, we also may set up a machinery to have such kind of structures. You have a flow up and a flow down, and you have some in-between layer, and the problem is how is, for instance, oxygen transported from the lung, side to the blood, and how is CO2 transported from the blood to the lung. And and it's also we have cooperation with the experimental group. And to do this, I will show you shortly afterwards. Then glomerular layers in the kidney, very, very important. The kidney is a very complicated filtering system. And I just show you, don't look at this part, just look, you have here a structure like here too. This is such a filter structure, and again there at this location a lot of ACE is placed, then ACE2, and here you have essentially an idea of how we try to do this, and we have already set up such an experiment. one thing represents the capillary space, one the air space, and you have here this layer, and in this layer you have really epithelial or cell systems are put, and we see how the exchange between the flows up and down is going on. It's done together with this group in Regensburg, and I could do this, let me finish with one question. If you had just such a system, you have a flow here and a flow there, and you have here a perforation there. What are the law describing the, what happens? How do we describe this system? A coupling of fluid flow with a mechanical system. And this is one, at least this we have to do. It's very complicated. What do? We try to say this is getting thin. The structures are also scaled, let me say, of the same scale. We assume here periodic one. What is a limit system if you have here elastic structure and you have the fluid flow? Also in the porous media, and there's nothing existing so far on that. There's something done without flow. What are limit equations of that? And there we especially have a problem. Here we would have up there Navier-Stokes flow or Stokes flow. Let's take Stokes flow here. Let's have here linear elasticity, but obviously Lagrangian situation. And now they are deforming because they are elastic. But then the problem is usually at the surface of the solid, we have transition, continuity of the velocities, and also in pressures balance. But these are nonlinear conditions because we describe it Euler and Lagrange on the solid side. We have to linearize this, and it's terrible for us. And it's terrible for us that we have to do it because it only gives small displacements, and but it's an obstacle for mathematics and also for the computational side. And I can tell you, new mathematics is needed. There, for all these studies, in order to make asymptotic analysis for that, you have to have a concept of scale-depending convergence. In mathematicians always need for equations estimates of the solutions. These estimates all contain epsilons in it. And then we have to find out, can we find some convergence, principle of convergence where we have a limit, scale limit. And this has been done. It's too complicated. If someone is interested, can show the slides. Let me just tell you what comes out. What is very interesting for us, essentially, we get out in the limit a plate equation coupled with a fluid flow. And this is a more complicated system. I don't want to disturb you at the end. I prefer a discussion. But if you are interested, it's a system where essentially for the normal move displacement, we get a plate equation. For the horizontal movement in the limit, you have here just a surface, a flat surface where we have the coordinate system sitting on. There is a second order equation, and they are is coupled with the elongation, displacement in the normal direction, and we have a coupling between the velocities up and down. And what we also could show is there exists a corrector term. Formally, we know the corrector term, which we would like to know because we want to see how much fluid goes through. So far, we are not able to say we have a fluid flow through through this membrane, through this plate of order, let me say, epsilon squared. Now, if epsilon is a scale factor, that's still a lot of mathematics to be done. Let me just skip this. I show you just shortly shortly the equation. I didn't plan to have this everything, but I would like to show you the equation. You have now, this cylinder is cut by one surface. I will be finished. One surface, and you see this is up. We see the velocity up. And here you have a u 0, 3. So it's an elongation in this. It satisfies this plate equation containing a u1, which is giving just a stationary equation for including the other terms. And this is a result which you can compute, and we have not computed this yet, but this is just recent results. And this is published in a memorial lecture for a volume for Andro Mikulic. If I can show you privately how one derives it, that's why I kept these slides in it. Now let me see. Finish. Current quantitative research in the life science relies on process-oriented approach, mathematical modeling, simulation with close integration of IT-derived analysis of real-world data, or computer experimented by computer experiment derived is advised, and should be a greater use should be made out of these results. So problems considered in these lectures lead to highly complex systems which usually represent sub-processes of different types and whose treatment cannot be accessed with standard procedures, as everything is really new. We are mainly have to contribute to systems reduction, because finally we want simple systems. But the main aim is watch out what is the important information which you have to keep to get it from the molecular level, which we have to take into account because we know data on this, to the upscale level. Then I want to finish. Oh, wait a moment. This was complex systems, high, even huge dimensional state spaces in multiple scales, stochastic space and time, coupling of multidisciplinary character. Process and data-oriented approaches have to be combined. Probably in research-based treatment of complex systems in general, but especially systems arising in bioscience and medicine is a very important topic of the future. I think only I want to quote, only those who know their goal will find the way. It's Lao Tzu said it. We have to know what we want, and we hopefully also find the way to it. And outstanding scientists such as Beltrami, Gauss, Riemann, and Klein, with whom he was associated, Beltrami, were aware of these facts. They really are excellent role models, especially for the young generations. Thank you very much.